Hi dear students. So our next session is on lactic acid production. Lactic acid is catabolic products of primary metabolism by microbes. It is also produced by many higher organisms including man who produces the acid in the muscle during work. Lactic acid products are formed from carbohydrate fermentation that are derived from pyruvic acid via the EMP, pentose phosphate and ED pathways. Products such as ethanol, acetic acid, 2,3-butanediol, butanol acetone, lactic acid and xylitol production can also be produced as byproducts. A lactic starter is a basic starter culture with widespread use in the dairy industry. For cheese making of all kinds, lactic acid production is essential and the lactic starter is employed for this purpose. The lactic acid bacteria. They are formicates group, non-spore forming and can be rolled or cocoa-shaped. Some of the examples are Canobacterium, Oenococcus, Enterococcus, Pediococcus, Lactococcus, Paralactobacillus, Lactobacillus, Streptococcus, etc. It lacks porphyrins and cytochromes. They do not carry out electron transport phosphorylation and hence obtain energy by substrate level phosphorylation. They grow anaerobically but are not killed by oxygen. They obtain their energy from sugars and are found in environments where sugar is present. The lactic acid bacteria is divided into two major groups, the homofermentative and the heterofermentative. In the homofermentative, it produces lactic acid as a sole product of the fermentation of sugars. Here, the glucose is catabolized into lactic acid alone. It converts the D-glyceraldehyde 3-phosphate to lactic acid by the Embden-Meyerhof pathway, that is glycolysis. Another one is a heterofermentative group of lactic acid bacteria. Besides lactic acid, it also produces ethanol as well as carbon dioxide, and it uses the enzyme aldolase. Aldolase is a key enzyme in the EMP pathway and splits hexose glucose into 3-sugar moiety. It catabolizes glucose into ethanol, carbon dioxide as well as lactic acid. The desirable characteristics of lactic acid bacteria as industrial microorganisms include the ability to rapidly and completely ferment cheap raw materials, minimal requirement of nitrogenous substances. It produces high yields of the much preferred stereospecific lactic acid and has the ability to grow under conditions of low pH and high temperature. Lactobacillus delbrueckii subspecies delbrueckii has the ability to ferment sucrose. Lactobacillus delbrueckii subspecies bulgaricus has the ability to ferment lactose, and Lactobacillus helveticus has the ability to ferment both lactose and galactose. Whereas in the case of Amylophilus and Lactobacillus amylovirus, it has the ability to ferment starch, and Lactobacillus lactis can ferment glucose, sucrose, and galactose, whereas Lactobacillus pentosus can ferment sulfite waste liquor. The organisms responsible for the production of lactic acid includes bacteria, Lactobacillus helveticus, Lactobacillus salivarius, brevis, viridescens, plantarum, Pediococcus damnosus, and the examples of fungi include Candida krusei, Saccharomyces cerevisiae, Rhizopus species. It requires only a simple medium and produces lactic acid, and it also requires vigorous aeration. Rhizopus species utilize glucose aerobically to produce lactic acid. Rhizopus species such as Rhizopus oryzae and Aryzis have amylolactic enzyme activity which enables them to convert starch directly into lactic acid. And this flowchart shows the normal lactic acid fermentation. The sugar is hydrolyzed to glucose and undergo glycolysis to produce pyruvate, and pyruvate undergo fermentation to produce lactic acid. Now let's see the lactic acid production by microbial fermentation and the important requirements. In this, the inoculum should be 5 to 10% of the liquid volume of the fermenter, and the temperature should be maintained at 35 degrees Celsius to 46 degrees Celsius and pH between 5 to 6.5, and it is maintained by adding base like ammonium hydroxide. Usually batch fermentation is preferred. Batch and fed batch fermentations provide high concentration of lactic acid, and continuous fermentation ensures high productivity of lactic acid. Airlift bioreactor is preferred when the organism used is Rhizopus oryzae. Rhizopus oryzae converts starch directly into amylolytic enzyme activity. Lactobacillus delbrueckii convert hexoses into lactic acid in fermenter at pH between 5 to 6.5 and temperature 46 degrees Celsius to 60 degrees Celsius. Lactobacillus bulgaricus convert hexoses into lactic acid in fermenter at pH 6 to 7 and temperature 42 degrees Celsius. Mixed culture of Lactobacillus brevis and Lactobacillus pentosus were used to produce lactic acid on large-scale production. Different substrates used for the lactic acid production. Carbon source. Carbohydrates derived from wheat, sugar, whey, cane, molasses, barley, and malt. Biomass of lignocellulose is a renewable carbon source. It is a low-cost and extensively available substrate. For example, sugarcane waste, banana waste, pomegranate waste, apple, peanut, husk, banana stalk, and wheat straw. Productivity is affected by temperature, fermentation time, and level of substrate. Highest yield is obtained after 7 days of fermentation by 18% substrate level is 7.76 gram per 100 ml, that is 77.6 gram per liter. Coming to the downstream processing or product recovery, the methods involved include ion exchange chromatography, nanofiltration, strong acid cation exchange resin, and foam separation technique is used. Now let's see the basic uses of lactic acid. It is used in the baking industry. Polymers of lactic acids are biodegradable thermoplastics, so it is used in the manufacture of plastics. In food industry as emulsifiers, and it is used as an acidulant, flavoring, and pH buffering agent or inhibitor of bacterial spoilage in a wide variety of processed foods. It is a very good preservative and pickling agent. Addition of lactic acid aqueous solution to the packaging of poultry and fish increases their shelf life. In medicine, it is sometimes used to introduce calcium into the body in the form of calcium lactate in diseases of calcium deficiency, and in the pharmaceutical and cosmetic industry, it is used as a formulation in topical ointments, lotions, and anti-acne solutions.